In this video, I want to present to you something known as single nucleotide polymorphism, or SNP, pronounced snip. It was given the name snip because imagine saying single nucleotide polymorphism over and over again can be tiring for the scientist. So the name that is snip was given. And I want to introduce it to you in a way that visually shows what a SNP really looks like. So single nucleotide polymorphism. Within this long name, single meaning one, and nucleotide is the four nitrogenous bases, ATGC, that make up our DNA strands. And polymorphism, poly means many, and morphism comes from the word morphology, which means form. So therefore many forms. Now I'm going to start off by giving you portions of DNA from 3 people. So let's say that this is a person 1, person number 2, and this is person 3. Remember, we have 2 copies of each piece of DNA, one from mom and the other one from dad. So let's say this genetic materials is from chromosome 2, and this is copy 1, so say from mom, copy 2 from dad. Now, at first glance, this 6 double-stranded DNA look to be the same, right? But my question for you is, can you spot something unusual or different in these 3 individuals' genetics? Given the fact that this set of DNA comes from the same exact location in the whole genome. So, in other words, these 3 genetic materials would have come from the same chromosome. For example, chromosome 2, and from the same locus, which is a specific location on the chromosome. After scanning over these genetic materials, you notice probably very quickly this line here. So CG, and this one's CG as well. TAN. Yep. So on this specific location of this DNA, we have AT and GC. But if we take a look at person 2, there is GC and GC instead of AT and GC. For person 3, however, the person also has an identical pair, but this time of AT and AT on either side of the 2 chromosomes. So now you can clearly see a variation, right, among these 3 individuals. And this variation is the kind of variation that is of one single nucleotide base, as in individual 1 having AT base pair here on the chromosome copy 1 instead of GC base pair. And individual 3 exhibits AT base pair on the copy number 2 of the chromosome 2 instead of GC base pair. We refer to a variation like this as a SNP. This is the definition of a SNP. The difference in a single nucleotide base among individuals at particular location in the DNA. Now, relatively, there are many SNPs in our genome. On average, they occur about once in every 1,000 nucleotides, which means there are roughly 4 to 5 million SNPs in a person's genome. And the vast majority of them are located within the non-coded regions of our DNA, which means these SNPs, these variations that we have between one another, are basically insignificant to us, or at least they are not our main focus because, for one, they have very minimal impact on our health, and that's largely due to them being far away from our genes. However, the other percentage of SNPs, the other small portion of SNPs are definitely something of our interest because they are located in our genes or near our genes. And as we all know, genes are necessary to create different kinds of proteins, right? And those proteins have functions that are critical to our well-being and normal functioning. And to give you a brief example, a protein called CFTR protein. This protein functions as a channel across the membrane of our cells that produce things such as mucus, sweat, saliva, our tears, and digestive enzymes. This protein transports negatively charged particles called chloride ions into and out of our cells. The transport of chloride ions helps control the movement of water in tissues, which is necessary for the production of thin, freely flowing mucus. And mucus is a slippery substance that lubricates and protects the lining of the airways, digestive system, reproductive systems, and other organs and tissues. So now, what made this protein? This CFTR protein is encoded by the CFTR gene. So CFTR gene. And in this CFTR gene, more than 1,000 mutations have been identified in people with cystic fibrosis. And this mutation in the CFTR gene, the ones that are referred to as disease-causing mutations, what they do is they alter the production and the structure or stability of the chloride ion channels. And all of these changes prevent the channel from functioning properly, which impairs the transport of the chloride ions and the movement of water into and out of our cells. So as a result, cells that line the passageway from the lungs, the pancreas, and other organs produce mucus that is abnormally thick or sticky. This sticky abnormal mucus blocks the airways and the glands, leading to the characteristic signs and the symptoms of cystic fibrosis. Now, a lot of these mutations in this CFTR gene are a change in a single protein building block, or one amino acid, in the CFTR protein. Remember, amino acids are the building blocks of a protein, and each amino acid is coded by 3 nucleotides. Now, going back to the main topic of SNP, which deals with a single nucleotide base, you can imagine the uses of identifying possible SNPs within genes or even in the regulatory region of genes. Each one of those SNP has the potential to alter gene function by way of created a different amino acid than usual. And as a result, created a protein that has a different structure. By example, this CFTR gene. Recognizing where the SNPs are within a gene or near a gene can give us important information or clues, such as whether some people are more susceptible to a particular disease or even our eye color. Although not perfect due to the nature of multiple genes involved in eye color, SNPs have been used as a predictor for whether you have brown, green, blue, or hazel eyes. In fact, based on a recent 23andMe data, it gets it right 80% or more of the time. So that's pretty amazing. Anyways, this is meant to be just a quick introduction to SNPs. I hope this video gives you an idea of what SNPs are, and you can see and imagine how SNPs are an important tool in biomedical research as well as in other research fields in biology.